And if you look at the last
So when we did this recording, we found that there were no major differences there in major aphid feeding behaviors except for one, phloem salivation. Salivation is when aphids are secreting salivary effectors into the plant, and we found that salivation is higher at around just as soon as the lights go off during the night. But even though salivation is higher, aphid consumption of phloem sap throughout the 24-hour period stays the same. So we found that there is a diurnal rhythm in salivation which peaks around the dusk, just after the light goes on, but there was no differences in phloem ingestion. So our question was, why is there more salivation occurring at night, and how does this allow the aphid to feed phloem salivation? So keep that in mind. Then we also wanted to figure out whether aphids, when they feed, they produce a lot of honeydew because they consume a sugar-rich sap. We wanted to understand whether there are rhythms in honeydew production. So we counted the honeydew droplets at the end of the day and at the end of the night. And we found that when the aphids feed on the plant, they produce a significantly higher amount of honeydew during the night. And when we put them on a diet where the food quality of the food does not change, at the end of the night they produced more honeydew, which means that for our paddy, they produce a lot more honeydew during the night, even though they consume the same amount of phloem sap at both times, during the day and night. Okay, so the conclusion from this first part of the work was that aphids have rhythms in both feeding and honeydew production. Next, we wanted to check whether they have rhythms in gene expression. So we collected aphid bodies at these different time points and did RNA transcriptome analysis. We found that there were two bimodal peaks in distribution. This Rose diagram over here shows the number of different genes that are differentially expressed at different times of the day. We found that they have one peak occurring around noon and another peak occurring around midnight in gene expression. And then we also clustered the genes based on their expression patterns, and we found that there were 4 different clusters. These clusters basically show that these aphids are carrying out different activities at different times of the day. For example, in cluster 1, which peaks during the daytime, the main activities are mRNA regulation and protein production. And then cluster 3, which peaks during the night, sugar and amino acid metabolism is what seems to be occurring. So then we wanted to look at a few important rhythmic transcripts in the aphids. We looked at circadian clock associated genes, and we found that they peak at different times. This is a heat map showing the expression pattern at these different times. And these bars over here indicate amplitude, which means that they show the highest amount of change. The bigger the bar, the highest amount of change it shows between the day and the night. So the circadian clock genes are associated at different times, and we are currently studying these different genes to see how they influence various aphid feeding behaviors. Another interesting area that we looked at was sleep. So when we came across this data, our first question to ask was, do aphids even sleep? If they do, when do they sleep? So based on genes that are associated with sleep from Drosophila, we found that if aphids do sleep, they sleep around noon. 4 ZT to noon, that is 4 hours after the light comes on, is when they actually sleep. So this is another area that we're working on to see whether we can identify any patterns in the aphid body if it changes when they're sleeping. So then the other important thing we wanted to do was to look at salivary effectors. So this figure over here just shows the different locations at which the salivary effectors can be introduced into the plant. And the R-PADDY, it already has its salivary transcriptome completed, and it has 264 putative salivary effectors. We did a rhythmicity analysis for all of these effectors, and we found that 42 of these effectors were rhythmic, and they fall into these 4 different clades based on their expression pattern. Out of these 42, 43% show peak expression at 12 or 16 ZT. So that means they are showing their highest expression in the nighttime even when our EPG data also shows that they feed or they salivate more during this particular time. So this is the first work which shows that aphids secrete different blends of salivary effectors in the plants over at different times of the day. So then we thought we could, based using this data, that we would be able to identify novel effectors which have a high impact on aphid feeding. So we use the amplitude, that is the amount of change between day and night expression, and the membership score into each of those 4 grades to identify effectors. The black dots over here indicate effectors that were expressed at night, and the yellow dots indicate that effectors were expressed during the daytime. So by doing this, we identified 2 effectors, C002, which is the most famous aphid effector that has been characterized, and another new one in R-PADDY called E8696. Both of these genes are expressed highly during the nighttime. So we wanted to study these genes further. And for this purpose, we used, sorry. So for this purpose, we used a microinjection. We injected the aphids, as you can see over here, with 40 nanoliters of double-stranded RNA, and we observed gene silencing for more than 48 hours. You can see over here again, it was injected between the coxa of the second leg of the aphid, and this did not result in any mortality in the aphids. So after we did the silencing, we found that the E8696, which is shown in these blue lines over here, had a very high impact on mortality or survivorship when the aphids were injected with it, and they also produced much lower number of offspring. Then we also used these silenced aphids to perform feeding assays, and we found that phloem sap ingestion was not really affected, but when the salivary effector has been silenced, the aphids try to salivate more to see whether they could actually produce the effector that they need for survival. And we also found that honeydew production in the night for the silenced plant was reduced. It was decreased compared to when we just introduced a GFP control in there. So to summarize, we found that there were rhythmic transcripts peaking around noon at midnight. The secretion of effectors varied over time, with 43% peaking during increased salivation. And this is the first evidence of diurnal rhythm-regulated salivary effectors and their time-specific roles in feeding. So we hope that with this kind of knowledge that we're just beginning to understand in both pests and aphids, this will allow us to optimize agricultural treatments by timing them when they're best suited. And we believe that these insights from chronobiology can provide new targets for biotechnological approaches to control pests. And with that, most of this work that was done by my postdoc, Dr. Jinlong Han, a graduate student in my lab, Daniel Kang, and a master's student, Miva. And this work was supported by the USDA and the Binational Science Foundation from Israel. I have collaborators there who are working on the plant side of things. That is Dr. Veradzin and Yoshi Goldstein, her master's student. With that, thank you again for this invitation and this opportunity to speak to you, and I'd be happy to take any questions.
Thank you, sir, for your thought-provoking insights. Now the panel is open for discussion. Thank you, sir, for joining.
Thank you.
Hello?
Now we'll break for tea. Let's
Hello?
join back in
Wait
10
a minute.
minutes.
Excuse me, wait a minute.
Yes, sir.
Dr. Vamsi,
Yes.
great good morning to you. It's a night
Thank you.
actually for us, right? Okay,
Yes.
and thank you for honoring us. We are really thankful to you to give us a thought-provoking, dynamic speech today. So thank you for you and Punya for agreeing. Hopefully, Punya will be joining tomorrow.
I think she sent a recorded lecture.
Yeah. I saw that. I saw her response. Maybe
Yes.
possibly if she has some time, she may be sparing
Yes.
minutes with us. Right.
Yes.
Pardon?
Yes, she'll try to join.
Yeah. Okay. Thank you. Thank you for your all your support. You know, you are supporting our students. Really, it will be helpful for the future line of works also. So I have only one quick question for you. Whether these
Yes.
dynamics, you know, the diurnal patterns, whether it is coinciding with the plant's response?
Yes. So right now we looked at plant responses and we found that there are difference in responses, especially with transcription factors that are getting regulated. We identified some worky transcription factors which are expressed at the same time as the salivary effectors. So we did parallel work in plants and aphids, and now we are trying to integrate the data to identify genes that the salivary effectors will influence in the plant. So that work is ongoing, and we will get that information in the next couple of months.
Okay, thank you. Thank you. Have a great time. We will wind up. Say goodbye.
Yes, thank you. Bye.
Let's break for tea and we'll get back in 10 minutes. Thank you.